This is TWIS. This Week in Science, episode number 592, recorded on November 9th, 2016. In it together. Hey everyone, I am Dr. Kiki, and tonight on TWIS, we are going to fill your heads with Neanderthals, assassins, and chickens. But first...
Disclaimer, disclaimer, disclaimer. Ladies and gentlemen, step right up, step right up. The American Sideshow is about to begin. Right this way. Don't be bashful, don't be shy. There's no way to walk on by. Give the tent with the painted orange top a try. Why? The things you are about to see cannot be unseen. That which you are about to hear cannot be unheard. It will be over when it's over. It is guaranteed to leave a lasting impact you won't soon forget. And your complacency is the only price of admission. That's right. Just sit back, stay home, and assume for the best, because what could go wrong? Right behind the tent flaps, sirs and madams, you will find out soon enough. If you aren't in, you're out, and you're going to miss out big league. This way, right, that's it, 2 at a time, quickly now. That's it, everybody in. Hey, you there. What's the matter, friend? Saving your complacency for a rainy day? Wait, you're headed for the wrong tent. That's no sideshow. That's This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And good science to you, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We keep it coming week after week after week. What a great show we had last week in Baltimore. Wasn't that a great show?
That was so fun.
So fun. And the Maryland Science Festival is still going on. If you are in Maryland, be sure to check marylandstemfestival.org and look for events that are taking place near you, somewhere in the state of Maryland, until I believe the end of the week, or actually middle of next week or something like that. No, the weekend, this weekend. Yeah, things are going on and on and on. Science, science, science. And oh my, what a wonderful bit of carnival barking for the opening of the show today, Justin. Thanks for that.
Of course.
Yeah, got me. And then our show. This is, I think it's the most excited I've felt all day. So here we go. Let's get this show going.
Yeah, I appreciate, on the carnival theme, addressing the elephant at the carnival, if just for a moment. Now we can move on and enjoy an amazing hour-ish of science.
Science. And on this week's show, I have a lot of science news. I have stories about plants and their affinity for light, music, and how it all takes connection to make it work. And yes, I do have a story about our next president, because that happened last night. So, and there is actually science
Neanderthals!
News. Yeah. Justin, what?
Neanderthals!
You have Neanderthals? Okay.
No, I was commenting on, yes, and I do have Neanderthals here as well, as the earliest life on land ever discovered was discovered. Oh, I kind of gave up the whole story there. Well, there's details. And age-old question, how the chicken crossed the road.
How?
Yeah.
Oh,
yeah. Did it?
Or when?
Why? It's a combination of all of them.
Oh, all right. I can't wait to find out about that, because, this is a question people have asked themselves time immemorial, because, chickens.
And children.
And children. That's right.
Children constantly asking this question.
And Blair, what's in the animal corner today?
Oh, I have the spiders for dinner that I teased in Baltimore, but we ran out of time to talk about. I'm very excited about that one. I have horny lizards, and I have terrifying creepy crawlies.
Oh, I'm so excited about that.
Yeah.
Not nightmare juice. We need more of it.
Chock full to distract. It's a distraction.
Right. We just need more nightmare juice. Oh my gosh. Add it on. Pour it on. I just
Scare
need
me awake.
a big ice-filled cup of nightmare juice before I go to bed. Thanks a lot, Blair. Okay. Let's get into the show. I'm going to just get this done with before we move forward. Last night was, or yesterday was Election Day in the United States, and the United States has elected a new president of the United States, Donald Trump. And today, news has come out as to the individuals that he will be choosing for his cabinet and also as his transition team. And the transition team is very important because they kind of go in and do all the groundwork, all the research before the president-elect actually gets into office and people actually have to hit the ground running and do work. So they're the team that starts doing work before the term actually begins. So this is important because it's related to the Environmental Protection Agency. Trump has reportedly selected director of the Center for Energy and Environment at the Competitive Enterprise Institute, Myron Ebel, to head the transition for the Environmental Protection Agency. He has been called an elegant nerd and a policy wonk by Vanity Fair, according to an article in Scientific American. He's the chairman of the Cooler Heads Coalition. This is a bunch of nonprofits that, quote, question global warming alarmism and oppose energy rationing policy. And in a biography submitted, this is again, according to Scientific American, when he testified before Congress, which is something he does fairly regularly in relation to climate science issues. He listed his recognitions as being featured in a Greenpeace, quote, field guide to climate criminals, dubbed a misleader on global warming by Rolling Stone, and he was the subject of a motion to censure in the British House of Commons after Ebel criticized the United Kingdom's chief scientific advisor for his views on global warming. He has called the Obama administration's Clean Power Plan for greenhouse gases illegal and said that Obama joining the Paris Climate Treaty is clearly an unconstitutional usurpation of the Senate's authority. In 2007, he told Vanity Fair, quote, there has been a little bit of warming, but it's been very modest and well within the range for national variability, and whether it's caused by human beings or not, it's nothing to worry about. He's going to be heading the transition for Trump into his next term. So his term. So this is just a taste of potentially what we have to look forward to on the science front when it comes to science related to climate and how our government puts policy together. Story over. Silver lining, please. Yes. Is there a playbook for that?
Just because it was broached, and we'll move on quickly. Silver lining to that is Florida is pretty much sea level and marshed as it is, so it won't be long before it's no longer an issue. Secondly, I do feel as though for the foreseeable future here, this show will less be about scientific inquiry for me than it will be a vehicle of escapism.
I think that's great in the short term, but in the long term, we need to start thinking about how we as citizens are going to demand that this gets looked at, because it's clearly not front of mind enough. We need to, all around the United States, start to look at this important topic and make it clear to our officials that it matters.
Yeah. And in addition to this, there are other things that are related to science, education. Trump is looking at hoping to dismantle the Common Core standards, which science is not a part of the Common Core as it stands. There are separate science standards, but I would assume that those would kind of go part and parcel with the dismantling of Common Core. You'd probably get rid of national science standards as well. Education is a big part of what this show is about. Educating, inspiring inquiry, and inspiring curiosity. And so that is what this show will continue to do, regardless of what happens in Washington. Moving on into some more really interesting stories based on that inquiry. Researchers want to know, what can we do with CRISPR in relation to humans? CRISPR, the gene editing technology that we've been talking about for a little while now. Researchers at Stanford University have published a paper in Nature this week reporting, or actually they published a paper in Nature in October, and then there's another paper that was published by University of California, Berkeley. Both of these reporting success in using CRISPR to edit human cells. The paper that was published by the Stanford team corrected mutations underlying sickle cell anemia in human cells. They reported a 30 to 50% survival rate and correction rate in the cells that they edited, and these cells lasted when transplanted into the bone marrow of mice for up to 16 weeks. Co-author on the study said this week that they have enough preclinical data now with mice, so animal models, that they are hoping to seek federal regulator approval for a human clinical trial to test the approach. Dun, dun, dun. Yeah.
Designer babies, here we come, right?
No, not designer babies, but with the potential to, if you do a bone marrow transplant, you're dealing with hematopoietic stem cells that come from, these are stem cells that are they make the blood. They build your blood supply. And so if you can do a bone marrow transplant, take out the diseased bone marrow that produces the blood cells that have the sickle cell shape, that is part of the problem with sickle cell anemia, to a bone marrow transplant with cells that have been CRISPR'd with a fixed the mutation repaired, suddenly you have the opportunity to have a treatment. People with sickle cell anemia undergo bone marrow transplants and blood transfusions regularly as it is. So this is not, it wouldn't be necessarily anything new. And this could be a huge, huge help to people who are stricken with this disease. So very cool stuff. CRISPR. Let's hope it gets regulatory approval. That's pretty awesome.
Pretty soon it won't even need it. But this is the dream, though. This is the dream that science has been pursuing ever since the genome was really discovered. Is the ability to affect change at this level in real time, not warding this off for a future generation, but by actually being able to change a gene or two or affect the cell in such a way that it functions currently in a human as a therapy, not as a preventative for future generations or an altering of genes before birth kind of a deal.
Right.
This is the dream. This is a fantastic example of everything that science has been working on in this direction.
Yeah, so fingers crossed that, we can get trials moving in humans and that those trials are successful. That's something that it's so far successful in mice, but people are not mice. We always have to do human studies. And then my final story to get us going, Zika. We are still worried about Zika. It's making its way with mosquito populations into the United States. And it is endemic in areas around the world. It is a huge problem, especially when it comes to women and pregnancy. We do not have a vaccine yet. People cannot yet get vaccinated so that they would produce natural antibodies to Zika and be able to fight the virus off. But researchers at the Washington University School of Medicine in St. Louis and Vanderbilt University School of Medicine have identified an antibody called ZIKV117. This is an anti-Zika antibody that was collected as one of 29 that was collected from people who had recovered from Zika infection. So these people basically built up natural immunity to further Zika infection. So if you like a cold or the flu, when you're exposed to something, your body creates antibodies that can help your body fight future infection off. These people created anti-Zika antibodies. This one neutralized 5 Zika strains in the lab. Among those, they picked like one of the most lethal strains of Zika. They just pumped it into these mice, and they're like, okay, people don't usually die from Zika, so we're basically going to give a deadly dose of Zika, a deadly strain and a deadly dose to these mice, and then we're going to give them these antibodies to see what happens. And this antibody protected the mice from Zika. Not only that, they gave it to pregnant mice. The antibody protected the females and it protected the offspring of those pregnant females.
Nice.
Yes. One of the co-authors says, we did not see any damage to the fetal blood vessels, thinning of the placenta, or any growth restriction in the fetuses of the antibody-treated mice. The anti-Zika antibodies are able to keep the fetus safe from harm by blocking the virus from crossing the placenta.
Science works.
Yes. And so the idea here now is that we don't have a vaccine yet, but maybe, just maybe, we can use this antibody as a therapy that people can take in the meantime. So you're going to have to keep taking the antibody because you don't have it naturally in your body. You'll kind of have to, like a pill every day or a shot every once in a while, to kind of keep the levels of the antibody up in your body while you're pregnant. But this is something that could very well help to protect pregnant women and their unborn children from harm from the Zika virus.
That's great. Just like with your prenatal vitamin, take your Zika antibody.
Exactly. And it could also be something that, it's been shown now that Zika can get into the sperm, get into the testicles of men and kind of stick around there for a long time. So this is also something that pregnant women and their partners could also be treated with the antibodies as well, just to be safe. Yeah. Yeah. So they don't know whether these antibodies can clear persistent Zika infection from people's bodies, but they're right now they're working to get a better understanding of how ZIKV117 binds to the virus and actually inhibits the infection from taking place. And one of the great things about this also is that like with dengue virus, sometimes there are bad reactions between different strains and different antibodies. So there is a possibility that Zika vaccine could actually worsen symptoms in people who encounter the Zika virus later on. This happens with dengue virus already. We know this happens. And so this is called antibody-dependent enhancement. And it's been seen with Zika in a petri dish, but not actually in people in the wild. So with this particular antibody, it was just effective. There was no problems. All good. Yeah. So
Nice.
far so good. This is really exciting.
Nice.
Yeah. Go medicine. Go biomedical research. Let's do it. More. This is This Week in Science. Justin, what you got?
Oh, hey, Mike, I didn't even know I had a story coming up. So let's see. Yes, the age-old question. Often, no, I'm not going to do that one yet. I got another one because this is the top of the show. Hang on. Here we go. Neanderthals. That's it.
There we go.
30,000 years ago. They were there, gone. but always with us, right? With a bunch of us anyway. In the form of DNA, sequences that are scattered throughout our human genome. There's bits of stuff that we can trace back and say, that came from Neanderthals times. A new study by geneticists at the University of California in Davis illustrates traces of our Neanderkin slowly being removed by natural selection.
What?
Says here. Yeah.
Natural selection got rid of Neanderthals?
In our genome, yes.
Yeah.
On average, there has been a weak but widespread selection against Neanderthal genes, says Graham Coop, professor at UC Davis Department of Evolution and Ecology and Center for Population Biology, and senior author on a paper describing the work published in the journal Public Library of Science Genetics. Let's see. That selection seems to be a consequence of a small population of Neanderthals mixing with a much larger population of humans. Neanderthals split from our mutual African ancestor over 500,000 years ago. They lived in Europe, Central Asia, some I think even into Northern Asia, which is like Siberia-ish. Well, not Siberia, but just northern, like Russia, northern Russia areas. We've also found them there. Thanks to DNA samples retrieved from a number of fossils, we have enough data on the Neanderthal genome to identify their ancestry within our own gene pool. When the most modern humans left Africa 50, 80,000 years ago, spread throughout Europe and Asia, they interbred with the Neanderthal. The first hybrid child would have been about a 50-50 mix, human and Neanderthal genes, and could have themselves bred with more modern humans, more Neanderthals, or other Neander-human hybrids. So what happened to all of this Neanderthal DNA? Today, Neanderthals genes are a few percent of human genome of people with European ancestry. It's actually a little bit more common in people in East Asian descent. It's almost absent in people of African ancestry, with the exception of sort of swaths of North Africa where there was people who went back to Africa after the migration. So what happened to it? Coop and postdoctoral researchers Ivan Jurek and Simon Aschbacher devised methods to measure the degree. I've totally mispronounced your name, sorry. Devised methods to measure the degree of natural selection acting on Neanderthal DNA in the human genome. One hypothesis has been that Neanderthals quickly became genetically incompatible with modern humans. So their hybrid offspring were not fit in the evolutionary scheme of things. They either failed to thrive in the society or they were not fertile. However, the researchers found something different. Rather than a strong selection against a few Neanderthal genes, they found weak, widespread selection against many Neanderthal DNA sequences that it is slowly removing from the human genome. That's consistent with the small, isolated population of Neanderthals mixing with a larger population of humans. Interbreeding in small populations, which is what the Neanderthals were, genetic variants can remain common, even if it's not like a really good thing. You could have these sort of, negative genes or these genes that have drawbacks to them. But because you're kind of stuck in this small gene pool, they stick around. There's not an alternative. There's not a replacement.
That's exactly it. It's like everybody's kind of got this deleterious mutation already. So that's what the offspring are going to have. And it's not deleterious enough to just
kill everybody.
Not
to wipe out the whole group.
Right. It's just kind of just a little bit. It's like, it's a club foot. Your foot's just turned inward a little bit. It doesn't really.
Everybody's bow-legged in this town. It's just how we roll. Yeah. But when they mix it with the larger population, natural selection starts to act against those variants, weeding them out. It says here, human population size has historically been much larger, and this is important since selection is more efficient at removing deleterious variants in large populations, Jurek says. Weakly deleterious variants that could persist in Neanderthals could not persist in humans. We think that this simple explanation can account for the pattern of Neanderthal ancestry that we see today along the genome of modern humans. And this is always something that I've sort of disagreed with, although the findings are consistent with other recently published works. If Neanderthals have been more numerous when we first encountered them, we might have a different mix of Neanderthal and human genes. So
this is also very similar to like the bonobo and chimpanzee study we were talking about a couple of weeks ago, where it's, probably a small number of individuals mixing into a larger population, and then it just kind of goes away. But what were you going to say?
Well, the part of it that bothers me in this about what is it, I keep thinking, gosh, the Neanderthals must have had some very advantageous genes for them to still be in the numbers of 2 to 4%, depending on what population that you're looking at. Mm-hmm. Those must be extremely strong, because just the large mix of humans of generation after generation after generation, over tens of thousands of years, you could expect to see almost nothing of the Neanderthals. Or if it was there, we are making this argument that there were these genes are just slowly being removed. It doesn't necessarily have to be deleterious. It could just be overpopulation growth and mixing of humans on a planet. There's no reintroduction point for Neanderthal at this point. And it also does maybe indicate that even though it was a small population, the population of humans may not have had to be that much bigger at the time, except that there must have been a lot more, according to the way I'm reading this. There was a lot more inbreeding. It wasn't a 1 or 2 or 3 or 4 off. It was these communities, this small community of Neanderthals and this larger community of humans, which I can't imagine was that big, but I don't know what the numbers of population they're talking about here. Must have been a kind of common thing.
That's another aspect to it. Yeah, that maybe it was small populations of Neanderthals and larger population of human, but interbreeding was common. It became common, and maybe that's why there's so much left, even though they're not necessarily beneficial genes. I mean, we've talked before about these Neanderthal genes potentially being the cause of allergies or autoimmune disorders, and so there is a question as to why the genes did or did not get weeded out of our genes.
And again, if it's a smaller population, that entire population may not have gone extinct from starvation or lack of hunting grounds. These populations could have just been assimilated,
absorbed,
like an amoeba, a paramecium engulfing its prey. The humans took on the Neanderthals.
Resistance is futile because I'm kind of tall.
Assimilate or be destroyed.
Right. So there's a lot to learn here still, and so again, this continues to be a topic that fascinates.
Fascinating. Neanderthals will always be fascinating. Always.
Said the Neanderthal.
Said the Neanderthal. There we go. If you just tuned in, you are listening to This Week in Science. Hey, you guys, do you know what time it is? Do you?
I think so.
Is it bedtime?
Yeah.
Blair needs a nap.
No way. Blair's in charge now.
Oh,
it's time for Blair's Animal Corner.
Whoa.
She loves our creatures, cried as small. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels. That I'm no closer.
What you got, Blair?
Oh, well, let's get right into the nightmare juice, shall we?
Yes, please.
Yes. So let's talk about giraffe assassin bugs. Not only are they terrifying looking, but they have a very interesting way of catching their food. They eat spiders.
That's
so
good.
The spider dinner I was referring to earlier is eaten by the assassin bug, not by us. Sorry, guys. But the assassin bug, you might think that's a good thing until you remember all the ecosystem services that spiders provide. Then maybe it's not a great thing.
Oh,
Right. They're kind of important. But the way the assassin bugs eat the spiders is what makes them so nightmare-inducing and fascinating. They sneak up on spiders in their own web. They drive their beaks or their mouth parts or whatever you want to call them into the bodies of the spiders, killing them quickly by sucking out their innards.
Oh.
So that's the assassin bug. The giraffe variety has a long neck, and they sneak up on spiders in their own webs while spiders are waiting to catch prey. So we've talked a bunch on this show about how spiders catch prey in their webs. They mainly recognize when a fly or something else is caught in their web by the vibrations. So we've talked before about how spider silk is kind of like a guitar string, and they can pluck their own web to kind of communicate or figure out what's going on. They can listen to the web themselves. So how does the giraffe assassin bug sneak up on a spider in their own web?
That's crazy. That's not possible. No insect could be that agile, that fleet of foot, that whatever.
Well, Macquarie University in Australia has discovered the secret. How do they do it? They wreck up the place. So what they do, Fernando Soli from Macquarie University took spiders, he put them in the lab, and he encouraged them to spin a web. He aimed a laser at the webs and then released the assassin bugs. The assassin bugs stalked the spiders. He filmed the results.
The assassin
bug. And they found that the bugs actually used their four tarsi, the tips of their front legs, to grab the strands of web ever so carefully, pull them apart, tearing the web, and then ever so carefully release the strands, avoiding recoil, avoiding vibration, and then moved on to the next one in a straight line for the spider. The sensor revealed that the technique allowed them to go up to the spider without causing any ripples in the web. They also played with wind, so they had fans on varying levels in the lab at different times, and the assassin bugs greatly preferred when there's wind because it kind of masks what they're doing even more. They also varied the timing of their web tearing so that it could not be in any way rhythmic or anticipated.
That's sneaky.
Wow.
Sneaky little bugs.
That is an intelligent bug.
Yeah. So next time you tear down a cobweb, think about how an assassin bug does it. See if you can tear one strand at a time and release it. And one strand and release it.
I think this is my new favorite bug. Although I'm glad that they don't live on a more macro existence where like
knock. They're just knocking on you. Knock.
No, you wouldn't even hear them knocking. That's the thing. You'd just be sitting there on the couch watching television and it's like, oh, where'd my innards go?
Suddenly your house is gone because they've taken it board by board and you didn't notice. And
someone sucked out my innards.
Oh my goodness.
I'm dead now.
So the pictures of the assassin bugs in general, many different species of them. The question is this only the giraffe assassin bugs or is this all assassin bugs? Because I see assassin bugs, or I've seen them before.
Really?
Yeah.
They live in Oregon?
Yeah, I'm pretty sure I've seen them before.
Interesting. Well, the giraffe assassin bug is very weird looking. Its long neck, it has that super long neck, but then it has the four tarsi in front. So it's almost like their arms are up here and they have just like the longest torso. It looks very odd. I do know that different assassin bugs, go ahead.
They're also called kissing bugs.
Ugh.
So if usually you see them during mating season when there are two of them together. But
No thanks.
Yeah.
But I do think that different assassin bugs eat different kinds of spiders. So I'd assume, for example, if an assassin bug eats a funnel web spider or a trapdoor spider or a spider that does not use web acoustics to catch their prey, obviously they're not going to employ this technique. But I think that is definitely the next step in this process, is to now look at other assassin bugs that eat spiders out of their own webs and see if this is happening across the board, because then you can find this very complex adaptation much more basally in the like evolutionary history of these guys. Terrifying. Crafty.
Oh, I just found out more terrible things about these guys. They
Oh,
will,
hit me.
yeah, these assassin bugs, the Centers for Disease Control and Prevention are saying that moving through Central and South America, but now rising in southern United States, infections of Chagas disease, which is caused by Trypanosoma cruzi parasite, spread through the bites from the assassin or kissing bug. It bites around the eyes and the mouth when you come out to feed at night.
No, 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 no.
Yeah,
Kiki,
Chagas
no.
disease. Oh my gosh.
Around the eyes and mouth while you sleep. Oh, God. I already didn't sleep last night. Now tonight, what luck do I have? Well, let's forge on ahead, shall we?
Let's forge. Yes, please. Let's
To
forge ahead.
thorny devils.
Okay.
Talk
Thorny,
about them.
yes. Yes.
Thorny devils, aka thorny dragons, aka thorny devil lizards. They are known for being very spiky. They're also from Australia. It's like the Australian Animal Corner today. But their barbs actually don't keep us away. They're not venomous, nothing like that. It just makes them look like they really would not be delicious to eat. And they live in a few different habitats, but this species that they were looking at, the Aachen University in Germany, I think I said that right. They were looking at a species in Australia that lives in the desert. They wanted to know a little more about this animal's insane adaptation for drinking. A few years ago, researchers found how these animals drink. They don't drink normally because they eat ants, and their mouth is so adapted to eating ants that they cannot sip or lick water.
Huh? So then how would else you get your... Do they just live off the liquid within the ant?
Listen to this. They have...
I'm leaning into the mic like it's...
They have teeny tiny folds and crevices in their skin that overlap, that create a tube-like capillary system that carry the water like blotting paper up to the mouth. So there's this amazing video that I will share with you now. If I can figure out how to screen show. Oh, I think Kiki has it. Kiki has it. Yes.
I got it. Don't you worry. I gotcha. You just talk.
Okay. So this is insane. I've never heard about this before, but you can watch it move just like blotting paper from the tips of their toes as they stand in a puddle through their entire body up to their face to their mouth. And then if you watch them long enough water collects in the back of their throat from this capillary action that they swallow. There he goes. He's swallowing. So that's not enough craziness. The question now is, that works fine when you're standing in a puddle, but how the heck do you get wet enough to move enough water like blotter paper to your mouth when you live in a desert? So these researchers, again, from Aachen University, captured some specimens, took them back to the lab for study, and they tried allowing the lizards to stand on sand that had been wet. Some water moved up their legs into the capillaries, not enough to do anything. Eventually, they found out it was about this weird habit that the lizards have of pushing sand onto their back and patting it down onto their back. This means that over time, gravity pushes the water through the sand onto their back and then moves downward through the capillary action. Eventually, enough water reaches their skin where it moves to their mouth like a straw and into the back of their throat, and then they can drink.
Wild. That is awesome.
So I think there's something to be said here for the use of capillary action. Highly underestimated. You can have skin folds line up perfectly so that stepping in water brings it up, fights gravity to your mouth, and beyond that can pull enough water out of sand to give you something to drink.
So I wonder if there's part of that capillary action is just the folds pushing it along, or if you could actually be sort of like using your body as a straw.
Well, if you watched the video that Kiki shared, their head wasn't wet until the very end.
No, and it wouldn't be, but that's what I mean. But like if there's like a little bit of a sucking action that's going on.
So once their face is wet, then they can kind of encourage more movement. Is that what you're saying?
Well, it would be the causing of pressure, I guess, like how a straw works.
So I would say it would be more likely that there's some sort of charge, because water is fairly electrically charged. I don't know if there's something going on molecularly that is allowing the water to move faster. I would say most likely it's just physics. It's just the fact that these channels are moving, are perfectly aligned so that the water is suctioned, just like blotting paper, to the mouth. And
Or
it's just
capillary action is also used in plants. It's how water goes from the roots to the shoots.
And it's
very common.
interesting too, because if you look at this
capillary
thing,
action is how straws work.
yeah, if you look at how
fair point, Kiki, +1.
Wait, that was my point. Hang on. If you look at this thing, for those of you who are just listening, this thorny devil dragon lizard looks like it's covered in kind of like rose thorns.
Yeah.
And that's also sort of an interesting thing for a desert creature. That's, there's lots of prickly stuff in the desert. The desert there's just not a lot of life in the desert.
But
what
actually,
is there just
that's
doesn't
totally false, and that is a very common expectation for the desert. The desert is teeming with life.
Yeah.
The reason that everybody's so spiky in the desert is that blood is water. And so the easiest way to get water in a desert is from eating other living things.
And also, I think
Okay.
the
Yeah, fair point.
spikiness, it's not the same as it does add surface area, but at the same time, it probably I'm wondering how it affects evaporation. and the
That's a good
loss of
question. I think
water.
it would depend on what the spines are made out of. So if they're
Yeah.
just made out of keratin and they're not a part of this capillary network,
Yeah.
it wouldn't, in theory, affect it very much.
And I also wonder if this capillary action would work on a larger animal. This is not necessarily the thorny devil fits in the palm of your hand. It's not like a giant Komodo dragon,
Right.
Right.
And it's also very interesting because it's in a reptile. Reptiles have watertight skin. This would
Yeah.
not work so well in a mammal that has semi-porous skin, for example. It would all just get sucked right in, and that would kind of defeat the whole purpose.
And
So
then
that's
you'd look
why,
like the Stay Puft Marshmallow Man.
right, exactly, just a giant sponge.
That's right.
But that's why I found this so fascinating is I had to read the headline a few times because reptiles can't absorb water through their skin, and that's what it sounded like was happening, and I had to dig deeper. And actually, they're just moving it over their extremely water repellent skin
which is perfect
towards
for
their
this.
mouth. Yeah,
Yeah.
it's perfect.
It's just a directional straw. That's
Yeah.
like Justin said.
It's just like
Yeah,
he's
that's
just
the way to go.
covered in straws.
That is so wild, so cool.
One wonderful straw. It is so cool. You know what else is cool? Just science in general. And
True.
It's cool to be enthusiastic about science because there is so much to learn. This is a big, wonderful universe. And we have more science coming for you in the second half of This Week in Science. Stay tuned for more after this.
Can't explain the things you heard with more than intuition. The line of reason shows the way to go. No conclusion.
The methods and the hypothesis, I hope they lead you to TWIS.org. Did you know if you go to TWIS.org right now, you can pre-order the 2017 calendars, which are still in production. Blair is finishing up the artwork. Our friend Patrick Harnett from Baltimore, Maryland, artist there, is helping to finish the calendar with us. And we are very excited that we can offer this calendar once again with science holidays, reminders to watch TWIS every Wednesday, and all sorts of beautiful art from Blair. We're very excited about this. So TWIS.org, there is a link, pre-order for 2017. Get your orders in so that you can get one of the 2017 calendars. Be the first. Send it as a Christmas present, a Hanukkah gift, whatever you like. It's amazing. Yeah, or 5. You could order 5. We're not going to limit you to 1. You can order as many as you want. Exactly, Blair. Thank you very much.
Put them in people's stockings.
That's right. Calendars that go in stockings. Additionally, if you head on over to twist.org, you will be able to check out our Zazzle store. Our Zazzle store is filled with all sorts of, look at it, there's that main header bar, Zazzle store. You click on it and it'll take you right to Zazzle, to the This Week in Science store, where you can get hats. And we saw Greg Guthman, I believe, wearing a hat last week. Thanks for showing up and bringing your hat that we all signed to the live show in Baltimore. That was pretty awesome. There are mugs, t-shirts, sweatshirts, stamps, cell phone covers. There's original art by Blair. You can get a tie. You can have Blair's art covering of T-Rexes on a tie. Like, if you want some science art to wear, this is where you go. Our Zazzle store, a portion of the sales goes back to support TWIS. Additionally, if you'd like to support TWIS financially, we do have donation buttons on our website, twis.org. Click on the donate button. That will take you through a easy to fill out PayPal donation form. You can choose one-time or recurring donation, and at any amount that you prefer. Also, we are using Patreon as another way. So if you click on the Patreon link, it will take you to our Patreon platform, our Patreon account, which is patreon.com/thisweekinscience, where big button, become a patron. You click on it and then you choose your amount, whether it's $1, whether it's $10. If it's $10, you can do a Google Hangout with us, with me, with whoever else can make it once a month at least. And then $15 or more per episode, and you're going to get a hand-drawn picture from Blair. One of the pictures that is in the calendar, but you get the original art made by Blair for you or for a gift, whatever you prefer to do. Makes it super easy. The main place you go, twist.org, to be able to find all of this stuff. And if you aren't into the financial stuff, if you don't have a lot of money, if you're not really able to buy things or support us on an ongoing basis, the biggest thing that you can do is to tell people about TWIS, because the more people we have listening to TWIS, watching TWIS on YouTube, the better it's going to be for everyone. The more we're going to be able to bring in donations to be able to do more for you. So help us produce TWIS either by supporting us, buying our merchandise, or donating to Patreon or through PayPal, or supporting us and producing by telling people about it. Help us with marketing. Tell people about TWIS. This is what we need.
Now more than ever.
Now more than ever. We thank you for your support. We really could not do this without you.
The ramification of treatments from holy men leaves me slightly queasy deep down in the abdomen, convinced that the lives that they lead need adjusting. They drive to the bookstore and blindly start trusting the miracles and cures all laid down in black ink, never even bothering to stop and think.
And we're back with more of This Week in Science.
Oh, yeah. Hey, Justin, what you got?
An age-old question often asked by children, as we discussed, why did the chicken cross the road? And no matter what answer you give a child, they will come up with a different response.
I mean, isn't the answer is just to get to the other side?
At first, but then
Right.
it just starts there, and then it goes on
Okay.
forever. But it is also the subject of a recent scientific study. The question has been broadened a bit to include not just why the chicken crossed the road, but when, where, and how. Chickens are everywhere on our planet today, but that was not always the case. Main wild ancestor of today's domesticated chicken is the red jungle fowl Gallus gallus. It ranges from sub-Himalayan northern India to southern China and Southeast Asia. And was first domesticated about 7,000 years ago, give or take about 1,000 years, somewhere in that range. In tracking archaeological finds of domesticated chickens, researchers occasionally find clues to an ancient world of agricultural trade. So they can sort of tell, they know the origin point of the domesticated chicken. So when the chicken starts to pop up somewhere else, you go, aha, there was some sort of connection from this place to that place. In this case, the discarded bone of a chicken leg, still etched with teeth marks from a dinner thousands of years ago, has provided some of the oldest known physical evidence for the introduction of the domesticated chicken to the continent of Africa. Research from Washington University in St. Louis has confirmed this. Based on radiocarbon dating of about 30 chicken bones unearthed at the site of an ancient farming village in Ethiopia, findings shed new light on how the domesticated chicken crossed ancient roads and perhaps ancient seas to reach Africa and eventually every other corner of the globe. Our study, Cody voice, provides the earliest directly dated evidence for the presence of chickens in Africa and points to the significance of Red Sea and East Africa trade routes in the introduction of the chicken, says Helena Woldakiros, lead author of the postdoctoral anthropology research in arts and sciences, Washington University. Arrival of chickens in Africa and other routes by which they both entered and dispersed across the continent are not really very well known. This is one of those things we're still just figuring out. Previous research based in representations of chickens on ceramics and paintings, plus some bones from other archaeological sites, suggested that chickens were first introduced to Africa through North Africa, Egypt, the Nile Valley, about 2,500 years ago, which is, that's a sort of a landy route. That's kind of what you would maybe expect. The earliest bone evidence of chickens
as opposed to sea chickens?
Most of them flying there on their own. The earliest bone-based evidence of chickens in Africa dates to the late 1st millennium BC from Egypt, approximately 685, 525 BC. So what is that in? It's about 25. Yeah, that's the 2,500 years ago. However, this study published in the International Journal of Osteoarchaeology pushes that date back by hundreds of years. Yeah. So some of these bones, they did the direct radiocarbon dating, and they found that it was popping around 819 to 755 BC. So that's 2,800-year-old chicken bones that they've discovered here. Despite their widespread modern-day importance, chickens' remains are found in small numbers at archaeological sites. So again, not a whole lot of evidence for this. One of the tools, I think, I always find this very interesting and important. They did linguistic studies of ancient root words for chickens in African languages, which by themselves suggest multiple introductions to chickens to Africa following different routes. So from North Africa through the Sahara, West Africa, and from East African coast to Central Africa, scholars have also demonstrated the biodiversity of the modern day African village chickens through genetic testing of those chickens. So a combination of tacts, but if there is a sort of Arabic root word for the chicken, well, wow, this chicken might have was introduced by this sort of a trade route or connection between ancient peoples. But it says here it's likely that people brought chickens to Ethiopia and the Horn of Africa repeatedly over a long period of time, over 1,000 years of being introduced, as Boldekiros. Our archaeological findings help to explain the genetic diversity of the modern African chicken, resulting from the introduction of diverse chicken lineages coming from early Arabian and South Asian contexts and later Swahili networks. But it says here, our study also supports the African Red Sea coast as the early route. So these chickens, it may have come still across land. It may have been, as part of what they also linked this to was some marine activity. Some early mariners were active about the time these chickens showed up. So they could have showed up on boats 2,800 years ago. And you've gone, we couldn't hear you. You've gone mute there, Kiki.
I was saying it could have been sea chickens.
It really could have been sea chickens. And part of it might not necessarily have been a trade route per se, but if you're going on a long trip and you bring your chickens with you, you have, this is an easy way. This is before you could preserve food very well for these long trips. So you'd want to bring something that was living and also small enough to be portable. And then they could breed more chickens. They could produce eggs while you're not eating the chickens. Like there's a whole host of benefits for traveling with chickens if you're not allergic to them.
Well, I mean, the easiest way to keep meat from spoiling is to not kill it yet.
Not have it be dead. Yeah.
Yeah, that's true.
That's fantastic. All right. Let's take it further with birds in the sea. Yes. One issue with birds and the ocean, and this is exciting because earlier the Neanderthal study was a UC Davis study written by Andy Fell, who I know very well from the UC Davis media office. And this is another UC Davis study from this week is just loaded with it. I'm so excited. From one of my advisors in graduate school from her lab, Gabrielle Nevitt. One of her graduate students now, Matthew Savoca, is working on this is something that's been an interest of Gabby's for years, is the sense of smell in seabirds. And Blair, remember we were talking like a week ago and you were like, wait, birds can smell?
Yeah, I've been, well, I knew that some types of birds could smell, but it has been the conventional wisdom quotes for a long time that most birds cannot smell.
Well, most birds can smell. In fact,
turns out
birds can smell just the normal smells. And I think I've mentioned this before. It's a misnomer because when we used to do brain research with birds, the olfactory bulb would just kind of break off. It's not connected very well to the rest of the bird brain, and it would just break off. And people just thought they didn't have an olfactory bulb, when in fact it was just left behind in the brain case with the rest of the tissue in there.
I can't believe that.
Anyhow.
So just a quick note though. If you're removing a brain from a brain case, make sure you go back and check the brain case very carefully for any bits that may have been.
Hot tip. You heard it here first. Always check the brain case.
There you go. Well, in this particular case, we do know that seabirds use scent, and they are very, very amazing at picking up scents across the ocean. And in fact, Dr. Nevitt has been a big part of showing us that seabirds are able to pick up the scents of the compounds that krill give off. So the oils that krill that krill emit that comes out of their skin or their shells, it soaks up through the water and creates like an oil slick on the top of the water. And then that oil slick vaporizes and there's a plume of krill scent that seabirds can follow to find patches of krill in the open ocean. Kind of like moths and pheromones to be able to find their mates. The birds fly across the ocean, zigzagging until they come and find a plume of scent that they can then hone in on and be able to find a bunch of krill underneath the surface. They can dive down and eat it. One thing they also eat is a lot of plastic. And some species of seabirds eat a lot more plastic than others. And so Matthew Savoca, graduate student in Gabrielle Nevitt's lab, and Gabby Nevitt and another graduate student have published an article in the November 9th journal Science Advances. And it explains why they eat more plastic. Why do these birds just keep eating plastic from the ocean?
So I thought it had always had to do with their bright colors and shininess.
Yeah, nope. It's not the bright colors and shininess, because that's not really what they're attracted to with the food that they eat.
Right.
It's the smell. It's the smell. And they discovered that they used plastic beads that are made of the three most common types of plastic debris: high density polyethylene, low density polyethylene, and polypropylene. And they threw them into the ocean in Monterey Bay and Bodega off the California coast. These were in mesh bags so that the beads weren't just floating around in the ocean, and they could collect them again. But they could also see what the birds were going after, and they found that the plastic actually smells of a sulfur compound called dimethyl sulfide, DMS, which is a Q released by algae and other organisms in the water that coats the floating plastic. And DMS is a scent that like krill give off. This is one of the scents that the birds use to find their prey.
Darn.
And so the algae cover the plastic in the ocean and give off the same scent as the food of the birds that the birds want to eat. And so this is why these seabirds that track the scent of DMS to find prey, according to this article, are nearly 6 times more likely to eat plastic than birds that do not track the scent of DMS.
Wow.
Yeah.
Well, add to the reasons plastic in the oceans is bad.
Yeah, plastic in the oceans is bad. Okay,
But-
and this is just this study is fantastic. It really gives us something to clue in on as to what's going on naturally, though, and why these animals, why these birds are eating the plastic. Maybe we can figure out some way to stop it.
Well, a great way to stop it is something good that came out of all of our results from yesterday. So far, it looks like California is banning single-use plastic bags statewide.
Woohoo!
So that was
There you
something that, since this study was from UC Davis in California, and they did the study in Monterey, where the Monterey Bay Aquarium was very heavily talking about the plastic bag ban. That is great news that it looks like this is actually going to go through, because the easiest way to stop birds from eating plastic is to put less plastic in the ocean.
There you go. Let's do that. Exactly. Yeah.
But does that mean I can't get my plastic bags for my produce at the supermarket? Like when I'm...
There are all sorts of caveats, but you can't
That's fine.
get
As long
your
as I have those.
single-use takeout bag.
That's fine.
And
We've been
the produce bags and this kind of stuff, they're trying to move to biodegradables. There's all sorts of stuff going on.
And that's the key thing that I think that the other lesson that we can learn from this perhaps is that there might be a way to eliminate this molecular scent from the plastics in some way. Like we could, this could be another ingredient too, or thing that you take out of the process of manufacturing the plastics in the first place, so that we can continue to litter, because humans can't be trusted with a thing. that will make it less attractive or make the scent less,
I don't know. That's such a dangerous game to play because if you're adding more chemicals to plastic, that will also end up in the waterways. And we don't know what those results are yet.
Yeah, preferably removing something or augmenting the process. It might be
It's more
possible.
just knowing that all of this plastic in birds' stomachs is not a fluke, and it's not birds being stupid, and it's not all of these other things. It's the fact that we're putting something into the ocean that doesn't belong, that smells like their food. Let's not do that.
Agreed. But knowing that most people won't heed this, it's also
Justin, that's defeatist. We can talk more about this later, but that is a defeatist attitude.
Yeah, let's take this conversation to the after show, which if anybody watches our YouTube channel, our after show is after the show, and we have discussions about things that don't fit within the regular confines of our podcast. So let's move on and keep the stories coming. So Justin, you teased this earlier. Earliest life on land ever?
Yeah. Well,
what?
Yeah, that's what it is. So, team led by Semi Naben and Free at the Freie Universität Berlin studied from South Africa's Barberton Greenstone Belt. The rocks in the study are some of the oldest known on Earth. Not the oldest, but some of the oldest. They're near the oldest, with their formation dating back 3.5 billion years. In a layer that has been dated at 3.22 billion years old, tiny grains of the iron sulfide mineral pyrite were discovered that show telltale signs of microbial activity. These signs are recorded both in trace element distributions as well in the ratio between the sulfur isotopes 34S, 32S in the pyrite. Using instrumentation installed in PostM in 2013, the scientists showed that the fraction of the 34S in the core of some of the crystals differ characteristically from that of the same crystal's rim, indicating that the exterior of the grain involved a processing of sulfur by microbes, also called biogenic fractionation. So the composition of the rock, the shape of the crystals, the layering visible in the field all indicate that it was derived from an ancient soil that developed on a river floodplain 3.22 billion years ago, which is giving us the date of when this thing would have been sort of fossilized, I suppose you would say. Field data collected during the study imply that braided river systems transported the sediment containing the iron sulfide crystals. It is interpreted that the microbes living in the soil at a level that was continually shifting between wet and dry conditions, subsequently producing the rim overgrowths on the pyrite crystals. Based on this evidence, scientists conclude in their publication in the journal Geology that they have found evidence for biological activity on land at this very early date, and their research then pushes back the date for the oldest evidence of life on land 300 million years earlier than previously documented.
Wow. Nice.
So, couple of little caveats.
Yeah, there should be.
Couple of little caveats in here. Oldest life on land, but it was from a riverbed. It was maybe wet and dry, but this was also water was necessary, and it was probably submerged a lot of the time. So whether you want to count this as life on land or not, this is probably as good as it gets and as close as we can come to that at this point. It is not the oldest evidence of life, though, on the planet. As we reported in the past from Western Australia, there are signs around 3.5 billion years ago. And in Greenland, very recently we talked about this, 3.7 billion year old stromatolites, which are even more direct evidence of microbial activity. But because of its location on wet drylands, this is being tagged as the earliest to have survived on land, as opposed to rock formations in the sea.
Ah, okay. So that's the specific of it.
That's the little caveat. This one is on land. The other ones we believe were microbes that were undersea at the time of.
Got it. Fascinating. So. Cool. Mm-hmm. It gets older and older.
So first life on Earth, not flippers, not plants, but of course our friendly.
And not really that long after, I mean, yes, it's billions of years ago, but not really that long after the oldest life got started in the sea.
No,
You know,
no,
3.7 to 3.5 billion years ago to 3.2 billion years ago.
yeah, it's
I mean,
already on
that's-
land.
It's not that not that
So
far.
bacteria did pretty well back then, and the Greenland formation, rock formation, but with this one was 3.5 billion years old. Although the part that they dated is 3.2, but the formation itself goes back as much as 3.5. The Greenland formation went back 4.1 billion years ago. That's, I mean, the Earth is just over this in age. So pretty much as soon as there was planet Earth, stuff was living. And granted, the exceedingly difficult part of this is that there are only portions of rock that date back that far that we have access to that we can actually even study. There are these little places, a few points, right? We got Greenland, we got Australia, we got this area of South Africa. Very few places that we can actually study, and they have life.
But like you've said before, it's the life got started very soon after the conditions that it could start in were available. It just goes, and as we're looking outward into the universe, outward into our own solar system, you look out and you go, there's got to be more out there.
It's almost as if life finds a way.
Yeah, life does find a way. But I think that's the amazing thing about it is we have very few places that we could even look for this evidence on the Earth, and when we go there, we find it. So imagine all of the places that are no longer accessible because the Earth turns over. There's all sorts of things that remove rock formations, subduction, induction, lava flow, things just getting covered by other stuff. And where we found, so this could have been global. I mean, this may have well been everywhere. We're talking Greenland, Australia, and Africa, right? These are separate points, very separate points on the globe where we find this. It's very likely that planet Earth was a microbial home almost immediately after its formation, which exactly, you're right, does have massive implications for the potential of life throughout the universe.
Absolutely. Other things that are just really cool, plants. Plants are all over the planet. That's our green planet, right? Yeah. It's pretty amazing. And we've talked before, Blair brought up the story about the invader plant that sticks its roots in the other plant's roots and sucks up, the plant's genes and uses those plants' genes to do, I'm going to do what I want. It's like the honey badger of plants. Well, plants in general might be doing a lot more than we think. And one thing that researchers at Seoul National University in South Korea have discovered using Arabidopsis. Hey, Justin, how's your Arabidopsis?
It's long since dead. It had a good run, but I have
a brown thumb. Okay, you tried. All right. Well, anyway, Arabidopsis thaliana is part of the mustard family, a little flowering plant used as a model plant to study genetic aspects of development and other aspects of plant growth. Anyway, something that people have looked at in plants for a while is they've got these receptors called phytochromes. And phytochromes are light receptors. Now, it makes sense when the phytochromes are found in the stems and the leaves, right? Where light can touch it. But they're finding phytochromes in the roots. And so they said, what are these phytochromes doing down in the roots? And what they discovered, they put these plants in the dark and they attached little light sources to the stem of the plants through an optical fiber. And they discovered that the stems of the plants act kind of like fiber optic cables and transmit light to the roots.
Oh, that's cool.
Isn't this awesome? Yeah.
The roots are just having a discotheque underground.
Wait,
yeah. So
why?
It's not like lots of light. It's not like you could see down there, or like other organisms could use the light to move around and do
Could
stuff.
it be
But
an antimicrobial thing, like
well,
using certain frequencies of light to get microbes to back off? Or
there could be aspects of that, but with the phytochromes, when they get triggered by the light, they produce a protein called HY5, which promotes root growth.
oh,
So it's like, hey, there's a signal from the stems of actual light going, hey, we're getting lots of light here. It goes down to the phytochromes and says you should grow and support all of our growth up here, because we're going to grow. Stems and the leaves are going to grow. Well, roots should grow to support that too. And so they engineered the plants to have phytochrome mutations in the roots. They discovered that HY5 production declined, and then they put mutations into the HY5 itself, and the roots got all tangly and stunted, and they didn't do very well. And so they found basically that with experimentation, they found that it wasn't chemicals. It wasn't anything like just glucose or sucrose or any plant chemicals leading to the root growth. It was actually this light signal. So stems act like fiber optics to the roots.
They evolved to use this information in tandem with their stems. It's genius.
It's genius.
Well done, Mother Nature. Bravo.
Good design there. Absolutely. Hey, you got some snakes?
Yes. So what is the creepy crawliest of scaries for you, Kiki? Are you afraid of snakes?
I really do not like snakes.
You do not like snakes. So
No.
do you ever see
No.
snakes where there are no snakes? You ever
No.
go, ah, snake?
No, I mean, it's not like, the recent Mexico flight, snakes on a plane.
Yes.
That really happened recently.
That really did happen.
wow.
I know that as much as I love talking about them,
I see spiders more than I see snakes.
I see spiders.
so I don't anymore. But when I lived in the foothills, and actually there were rattlesnakes around, I would occasionally like double check like
Okay.
a stick or something laying it, because that was my constant fear, having encountered a few, that I would get rattlesnake bit and die on the spot.
Yeah, that makes sense when you're in the area. Yeah, exactly.
It doesn't happen to me like around town. Okay.
Yeah. So new research from Nagoya University indicates why that might be, that we are spooked and see snakes or spiders sometimes where maybe they aren't. It's actually based in evolution. It's because spiders and snakes can be venomous. Again, I don't want to demonize snakes. 1 in 10 snake species are venomous, so not a lot, but they can be dangerous to us out in the wild. And so recent research looking at visual awareness of snakes in humans indicates that we might be more on edge, more prepped to recognize snake shapes than the shapes of other animals. So there's a tool called a random image structure evolution RISE, used to create a series of 20 images of snakes, birds, cats, fish, and they ranged from completely blurred to completely clear, from I think it was 1 to 20, yes. And they then asked subjects to view these images and in order of increasing clarity until they could identify what they were looking at. So it would start at 1, 2, 3, 4, and eventually you'd say that's a snake or that's a cat. And the algorithm made it unbiased and random, and they found that overall, the snakes were well identified between 6 and 8 on that
Holy
image.
smokes.
Yeah.
Looking at it myself, I would probably see a snake at around step 9 or 10.
Yeah, I wouldn't until 13, but I blame the colorblindness.
It's a black and white image.
Yeah, but everything looks the same. So, but those are the snakes. They're between 6 and 8.
It looks like a dirty image until 12. Step 12
like
to
a
me,
scrambled TV channel.
wow.
All the other animals didn't get identified until the 9th or 10th image. That might not sound like much of a difference, except that out of 20, that's a pretty big jump to consistently be that far ahead, needing more clarity to identify it. So the humans were able to pick out snakes, even in what could be dense undergrowth in terms of the scrambled picture. So their indication from this, in what they confirm as snake detection theory, is that the visual system of humans and potentially primates have evolved in a way that facilitates picking out dangerous animals like snakes. So I say next is spiders. It's my personal opinion.
Yeah, for me, black widow spiders. It's kind of the same thing, Justin, living in an area where there are rattlesnakes. It's like I grew up in the Central Valley. There are black widow spiders, and like the basement of my house I grew up in, many black widow spiders.
Basements,
Wood piles.
garages, they're all
Wood
over the
piles,
place here.
Black widow spiders. It was just constantly like, oh my gosh, that's the web. That's the spider.
It's that enlarged abdomen. You don't even need to see the hourglass on her. It's just this enlarged abdomen. That's why we have false widows, right? That's all you need to indicate, I don't want to mess with this animal. So I say that's the next step in snake detection theory, is spider detection theory.
Either
Get on
that,
it, science.
Either that or special snakes on a plane detection theory. I have a couple of quick stories. We've talked before about this phenomenon of people who are indifferent to music, who really just couldn't care less about music.
Yeah, I
just
remember this.
one way or another. So some researchers have looked into this a little bit more deeply, publishing in Proceedings of National Academy of Sciences. They used functional MRI scans to track brain activity in 3 groups of participants. Now, not huge groups, 15 people, so take everything with a grain of salt. But one group identified as indifferent to music. One group identified as normal, just music's fine. And then the third group identified as having intense pleasure from music. And none of them were musicians. None of them had been trained in music in order to reduce any bias that might come from it, from the study. They each subject was asked to provide 2 pieces of instrumental music, no vocals, that they found emotionally pleasing. And so this was kind of hard for the ones who were like, I don't really care about music. What do you mean? And so they supplemented this music by taking music matching from Spotify, and then they imaged people's brains while they listened to these songs. And then there were other activities that they could match it against that are traditionally rewarding as well. So the idea is that, okay, these people are indifferent to music, so they're not getting the normal brain reward, right? If you like music, that means that your brain is rewarding you. There's something involved in the reward system. So they had them do other things that were also fundamentally rewarding, like gambling. That's another nice thing to do. And they found that these people had lower blood flow in areas involved in the reward system of the brain when they were listening to music. But there were no differences in blood flow to the reward regions when they were gambling. And so they looked at the auditory areas of the brain, and they found that there's just there's no connection between the auditory areas of the brain and the reward system part of the brain in these people who are indifferent to music. Whereas in people who like music, there's definitely a connection, and people who are highly interested in music derive great pleasure from it. There was a very dramatic and quick connection between the auditory cortex and the mesolimbic reward system.
So is the indication here that if you don't like music, it's because it's hard, your brain is hardwired to not like music?
And so that's the question. They don't know causality because this is just a point in time in people's lives. And so we don't know whether or not it's, I don't think I like music. I'm going to be indifferent to music. And that changes the brain from that point forward, or whether these people are born with just no connection between auditory area and the reward area of the brain. So that's the next step. Is it causal, and in what way? Yeah. And then in a final story, just because, Soylent, right? We've talked about Soylent. We never actually tried it ourselves. This is the kind of it's like a nutrition shake kind of thing that it's for geek, it's the nerd culture nutrition shake, the meal replacement system. Suddenly it started making people violently ill.
Oh,
gastrically violently ill. And there's been just a lot of reports of it, just everybody it started, and all of a sudden just people reporting all sorts of trouble with this product. The company's been trying to figure it out, and they thought that maybe it was a soy ingredient because soy is a known allergen and can cause gastric problems. And so they're like, oh, well, maybe it's this soy blend, the soy protein that we're using. But what they've discovered is that it's not the soy protein, it's an algal flour that they're using. This flour is made of dried algae, Chlorella protothecoides, strain S106, grown in giant tanks by Terra Via Holdings, Inc., under the product line Algabia. Now, the thing about it is that it has been approved for use in foods by the FDA as of 2014, signed off as a generally recognized as safe ingredient, GRAS. But there were notes. The submission to the FDA, the authors noted participants in several small human trials of algal flour reported gastrointestinal upset, including nausea, cramping, and bulky stools. This is according to an article in Ars Technica. So it's possible that this algal flour, even though it has been identified as a generally recognized as safe ingredient, is really not all that safe, or at least it's safe, but just makes people's tummies really upset. Yeah. So very interesting.
I'm still confused that people are drinking Soylent. That's a good marketing technique to name your product Soylent. So
It is fun. Come on. It is fun.
I'm confused.
Yeah.
But
So
don't you wonder?
yeah, it could be this. The Terra Via Senior Vice President Mark Brooks insisted that the flour is safe, saying our algal flour has been used in more than 20 million servings of products, and we are aware of very few adverse reactions. In no cases will the algal flour identified as the cause. But it is a possibility.
Yeah,
that
but
it's
name
algal
of
flour.
food that somebody can't get like constipated on. Like it's sort of.
Oh,
Oh, so it's like food. You put food in our food. How dare you? This was supposed to be humans.
yeah, but it is a big question. They're taking all sorts of ingredients, putting things together in a novel way. Algal flour really is a fairly new addition to our protein. Our protein intake. It's not used very widely as an ingredient. This is new. It shouldn't necessarily cause problems, but we don't go around skimming algae off the top of ponds all the time. So,
Well, but then again, we're also talking about, like, as we keep learning, it may be just based on your microflora, and that's
yeah.
And so,
And we don't know
Yep.
why some people are getting sick with the Soylent and other people are not.
Right.
It's not like everybody who's using
Some
it.
people can't handle dairy. I mean, this is this thing. There's all sorts of reasons for people's reactions to food that aren't universal to all humans because we have different microflora, we have different genetic makeups, we have different things going on within our bodies. So if you eat it and it's been fine, keep doing it. If you keep having the constant poops or whatever as the adverse affect, stop eating that thing. That's how you figure this out.
But it's also, if it's something that this is a new product, the Soylent is a new product, and it's something that is experimental. And if it's not just one person reporting this adverse reaction, but many, many people, then they really need to look into it, because it's not just, hey, this one person should stop taking it. There's something wrong with the formulation. This is affecting too many people. They need to change the formulation. So, yeah. Well, any more stories, anyone?
I'm good.
We're all good. We got to the end of another quote unquote hour of This Week in Science. Congratulations, guys.
Time is relative.
Yes, very relative. We stretch time here. We have a time warping for our hour. Thanks, everyone, for watching the show. Thanks for being with us. Thanks to those of you who hung out with us at the Maryland STEM Festival last week. It was super fun. Loved being in Baltimore. Such a good time. Patrick, thank you for being a great host. It was really, really fantastic. But right now, I'd like to thank our Patreon sponsors. Thank you to Chris Clark, Paul Disney, G. Burton Lattimore, John Ratnaswamy, Richard Onemus, Byron Lee, E.O., Jared Lysette, Ulysses Adkins, Kevin Parachan, Andy, Keith Corsell, Jake Jones, Dave Friedel, Mike McGraw, James Randall, Eric Schwab, Bob Calder, Mark Mazzaros, Ed Dyer, Trainer 84, Brian Hedrick, Layla, Marshall Clark, Charlene Henry, Don Kamarechka, Larry Garcia, Randy Mazuka, Tony Steele, Steve DeBell, Haroon Sarang, Melissa Mosley, Alex Wilson, Jason Schneiderman, Rudy Garcia, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Richard Porter, Daryl Lambert, Rodney, David Wiley, Robert Aston, Felix Alvarez, Darwin Hannon, Cosmic Gypsy, Brian Hone, Orly Radio, Brian Condren, Pixelfly Marked, Nathan Greco, Hexator, Deborah Smith, Mitch Neves Flying Out, John Crocker, Christopher Dreyer, Ben Rothig, Sylvan Westby, Artyom Shuwada, Dave Wilkinson, Steve Mishinsky, Rick Ramus, Gary Swinsberg, Phil Nadeau, Braxton Howard, Salgid Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insom, Michael George, Russell Jensen, Mountain Sloth, Jim Drapeau, Tara Payne, John Maloney, Jason Olds, James Paul West, Alec Doty, Illumalamo, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie, Paul Stanton, David Simmerly, Tyler Harrison, Columbo Ahmed. Thank you for all of your support on Patreon. And if you're interested in supporting us, you can find information at patreon.com/thisweekinscience. Also, remember, you can just help us out by telling your friends about TWIS. Tell 2 people this week about TWIS. That would help us out immensely. If each of you who listen to the podcast or watch the podcast told 2 people and they started listening, our numbers would just jump. So don't underestimate your influence. On next week's show, once again, we're going to be back broadcasting live online at 8 p.m. Pacific time on twist.org/live. You can watch and join our chat room. So much fun in there. Hey, guys in the chat room. I hope everything's going well this evening. If you can't make it, though, you can find past episodes at twist.org/YouTube or just twist.org.
Yep. Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have an Android device, you can look for TWIS for Droid in the Android marketplace, or for just check for This Week in Science in anything. See, it's the TV, there's phones, computers, there's all sorts of Apple devices you can plug that into, and you'll find us.
You'll also find more information on anything that you may have heard here today. That's like show notes. You can also make comments, start conversations with other listeners. All of that lives at www.twis.org. That's TWIS.org.
And what can you do there? You can make comments and
I
suggestions.
just said that. You go
Yes,
ahead.
but you can do that there, but that's sort of like putting it in a time capsule and hoping somebody finds it. You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS, at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a plan to survive the next few years, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you have learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Yo, 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 baby pops. Hey, hey. Yeah, you know you can do it. We'll keep it pumping hard like I wish it would. You know, let's da-na-na-na, da-na-na-na. Da-na-na-na. All right, everybody. Ed from Connecticut is saying Minion Hangout tomorrow, 11:10 midnight on si.org. Scienceisland.org. Scienceisland.org/Science Island chat. You can hang out with Ed and Pamela in Australia. Pamela and Chelsea. Oh, sweet little girl. Thanks, everybody, for watching the show this week. I hope that we were an addition to your week, a distraction from things that you're not necessarily interested in thinking about right now. It's good. I'm glad that we had this time together.
Here, here.
Here, here. And I'm so glad that we had our trip last week to Baltimore. Everything went very well. Got the show all out finally. Fada has been doing a great job of helping us out with the social media side of things, which is something I definitely needed help on with traveling and flying and posting, and just having the help to do that was fantastic. Also, Brandon helping us from Infinity Networks, who's been helping us to stream to Facebook, and he totally took that over, was able to do it on Friday, which is, awesome, because Wednesday night is our slot and people put time aside for that. And then for him to be able to do that also on Friday was going above and beyond and is hugely appreciated. So Brandon, thank you so much for helping us stream to Facebook last Friday. Also, you guys in the chat room, you are fantastic. This is what I'm working on these days, gratitude.
Things that are great.
There are a lot of things that are great. And there are a lot of things that will continue to be great. So one of those things, I haven't gotten details yet on when it's exactly going to be, but January 19th, we are going to be in San Francisco at the Cal Academy of Sciences Nightlife for SF Sketch Fest. So we'll be doing a 40-minute show. Oh, good. There you are, Brandon. Thank you so much. And Identity 4 also on a weekly basis. Thank you for recording the show for me, because that is another huge thing. So I don't have to think about the downloading and the recording and the catching the stream or all things. It's a lot of things. There are a lot of moving parts to do this show. And Identity, thank you for doing that every week. I really appreciate that. And everyone for just being here so much. Yeah, it's amazing. It's amazing. You guys join us for the show every week. I love it. Thank you so much. Yeah, January 19th, SF Sketchfest. Super excited about that. I don't know exactly what we're going to do there yet, whether it'll just be us for 40 minutes. You already know what we're going to do.
I absolutely know what we're going to do. We're going to bring the science, and we're going to do it live, and we're going to do it awesome. Yeah.
You guys, look, I'm indoctrinating children with science fact.
Oh, look at you being an educator, science educator.
I'm talking about chemistry,
science
and also climate change.
Science.
Hey, what were we disagreeing about earlier?
We don't need to talk anymore about it.
No.
I feel like it's just going to end with a lot of yelling, and I'm sick of yelling right now.
Okay.
Ultimately, I think that single-use plastics are not an inevitability, and that actually it's really easy to make decomposing single-use plastics, and that it is actually a economically advantageous thing to do because we have the technology.
Yeah, but what if they still smell like Krell?
Well, they will break up in their gut or in the ocean before it becomes a problem, so it'll be fine.
That sounds a little...
Because it'll be made out of corn or potato or something else.
Okay.
Perhaps the algae themselves.
That would be that. See, now you're thinking outside of the box. That's it right there.
I've been here the whole time. Let me curtsy to you real quick.
No. That what? I don't know what that was.
I live outside of the box.
Oh yeah, that
So-
would be fantastic. And then it might even be like encouraged to litter. Like every time you litter, you feed a bird.
Well,
If we could just figure out a way to get ourselves back into the ecosystem in a way like that, like
if we're going to make the plastic out of algae, why don't we just blend that into our Soylent?
Well,
Oh, yeah.
No, that's a
Perfect.
great. But
That's
that's
a great
I love the whole concept of that. Of
Like you put your sprouts into the biodegradable algae bag and just plop the whole thing into the blender, bag included.
There you go.
It's like rice paper. It's part of the recipe.
It's easy to get reusable cloth shopping bags instead of plastic. I now am the proud owner of about 24.
Oh God, I probably own 100.
Because I have a closet full of them now. Just because it never I think I've talked about this before, but you go to the grocery store, you've got your bags, you're really happy that they were in the car. They load them up, but maybe you ordered too much, or you shopped a little more than you had bags, so you buy one more bag.
Mm-hmm.
And you carry all these bags into the house. And the next day you're out driving around, you're like, oh, got to do some shopping. You stop by, and then you realize you have no bags in the car. They're all in the house when you unloaded the groceries. You forgot to put them back into the car. And so now you're buying more bags. And I've done this enough times that I've got quite a collection.
Right.
And what I've noticed, too, is if you bring a variety of bags, like I'll throw the brands out there because why not? I've got the local market is the Nugget Market. I've got the Co-op Market bag. I've got the Trader Joe's bag. I've got like from different places. And I'll have like just an assortment, and I'll bring like, a whole bag of bags in with me and then throw them in. I've noticed that they always, whatever store you're at, tries to prefer their bag first to load up. Like they'll sort through, they'll rummage through your sack of bags and go, oh, okay, here's some Trader Joe bags. We'll use those ones first, and hopefully we won't get to the others. We'll really pack it full, and that Nugget bag won't be seen, or if you're at Nugget, the other way around. But yeah, and it's, they're kind of fancy now. They're artistically decorated, and it's a good thing. There's nothing wrong with this.
Oh my God. I keep writing Yabby, and autocorrect keeps changing it to Rabbi.
Yes, rabbi shells. We were going to make plastic bags out of rabbi shells.
Okay, anyway.
But-
No, I'm good at getting them to the car, but I'm not very good at remembering to take them into the store, or I will often not take enough into the store. So then what I do is I just forego the bagging process, dump all of my groceries into the cart loose, walk out. The security guard is like, hey, what are you doing? And I have to show him the receipt. It's fine. Then I get to the car. But I kind of prefer this because I feel like because grocery stores don't give bags freely anymore, the art of bagging groceries properly has been lost. I feel like even people who stand there and bag, that used to be a very fine science, like cans at the bottom, then frozen food, then eggs on top. It was bread on top, like very clear. But now I feel like because people are trying to stuff everything into the 2 tiny bags you brought, they just, all the rules are out the window. So I'm kind of happier to be able to like wheel everything out to my car and then take my sweet time packing my groceries exactly how I want them in the bags, because otherwise you're rushed too when you're in the line and stuff.
So bad bagging is a pet peeve of mine, and I do trace it back to like I must have been 5 or 6 or 7 years old. I don't know how old I was. I was very, very young though, and one of my uncles or somebody, it was at my grandmother's house. I found this book on on that was like a training manual on how to properly bag groceries. And it was very detailed. Like it was a manual, and it was only about bagging. But it was like thick little manual on how to do this properly. And I remember like finding this and reading this book that somebody in the family must have like gone out for this job and gotten this manual, and that's why I was in my grandmother's house. But I remember reading, and so it's always stuck with me, which is again the importance of early introduction to children on subjects, on concepts of learning. Why it is so important that children, what you were doing, exposing them to elements of scientific theory. Because things like that stick with you for a lifetime. They really can take root. And so I've got a bagging pet peeve. I'm always rating my bagger's ability to have assessed the groceries that I've brought. Even to the point where I'm trying to help them out. By like if I've got canned goods and soft goods together in my cart, I always try to put all the heavies on the little conveyor belt first. So that even if they just do it in order of what showed up first, the order of what goes in to the bottom of the bag is correct. And I have to say, I would rate the Trader Joe's as being excellent baggers.
Trader Joe's are excellent baggers.
My
I would say.
Safeway baggers, excellent baggers as well. Typically,
Yeah, I'm one of those people who I also put items onto the belt in a particular order, hoping that they will bag them in that particular
exactly. That's why if you had your headphones off, this is what I just said.
No, that's what I'm saying. I do the same thing.
I do as well. Yeah. But I will say I'm going to call out. I'm going to do a little call out. Hopefully we don't get sued. Worst baggers I have encountered. The Target store. Like I almost don't shop.
Well, that's because they like barely even sell food. They're like food was an afterthought. But I agree. I've definitely had like my eggs at the bottom of the bag there, and then like the giant refill of hand soap on top of the eggs. It's like, what?
And I did like the grumpy old man sort of telling the kids to get off the lawn thing once. I really honestly had to I'm going to go over to the next station and it's okay. Like, are you? Yeah, I'm unbagging everything you just did and I'm redoing it because that was ridiculous what I just witnessed.
Oh, see, probably people hate me at these stores because I bag my own stuff. I don't even let them. I'm just like, nope, I got it. Thanks. No, go ahead, hand that to me. Oh, no, that's going to go in my purse. No, I got it. Especially at Target because you have the tiny items. I'm going to put that in my personal bag. Like, I don't want soap in with my food. I'm very particular. And some things I don't want bags for at all. Like, they try to bag laundry detergent. I'm like, this already has a handle.
Right.
So I'm okay.
Right.
I usually don't even let them bag.
So there is a socioeconomic layer to this too, though, right? I think there's an element of what you just said that slightly depends on how close your driveway is to your home.
Oh, and if you are even in a car, do you have a car?
Exactly. Like we were talking about this in the cab about like Treasure Island forgot to put any grocery stores and nobody put a grocery store there, but they put a lot of low income housing and these people have to, travel by mass transit.
Go to Oakland or
well there's the toll of which way they go to work, but there's also like they have to travel mass transit to get groceries. You can't just load up a trunk full of cars and take a couple runs of going back and forth. So yeah, there's all sorts of elements to this.
Brandon just posted in the chat room, nationalgrocers.org special program, Best Bagger Championship.
Oh.
There is actually there are videos you can watch.
Yes.
You can watch the final round of this year's competition. Maybe we should do that, Brandon.
Yes.
And I'm trying to think if it was something that was put into a movie or if I've actually watched a documentary on this.
I've seen it.
And
I've seen
movie, or is it part of an actual documentary on?
I think I saw it on, like local news when I was younger. I feel like they were like, oh, and young Tommy Johnson just won the best bagger contest up in Salt Lake City. Let's see how he won.
Yeah.
Thanks
I
for
think it's
that,
awesome.
Carl. I'm live in Salt Lake City with Tommy Johnson, who managed to put the cans at the bottom of the bag. Tommy, how did you get to be here today? How long have you been bagging? A whole 4 years since you were 13? That's fascinating. What do you, now that you've won, what do you plan to do? Well, I plan to go to the Disney store and bag everything there. Oh.
Blair,
It just
though,
got sad.
the fact that you bag your own groceries, do you think you could compete against these ultimate baggers? I mean, they're fast. Look at them. Look at them.
No,
They
absolutely
are
not.
They're choosing their items. They are bagging efficiently and quickly. These baggers they're intense.
Yeah, I don't think I could. I don't think I would
bag
be
my groceries.
judged appropriately. Also, because I'm more like, okay, all my frozen food, I just like literally throw it into a bag, and I'm like, okay, that's a bag, because it's all frozen. I want it all to be in there together. I also want to know. I'm very meticulous about the order I'm going to unpack my groceries. So usually I'm grocery shopping after work. I'm tired. I'm hungry, right? So I want my frozen food all in a bag. So I know to unpack that first thing, right? Then I know I have my bag of like refrigerated and vegetables and stuff like that. That's getting unpacked next. And then I have my bag of stuff that I could unpack in a few hours, tomorrow, whatever. And
Those
then I have my
ladies totally won. That was awesome.
Yeah. Nice. And then I have like my bag of like non-food, right? So that's more what I do is I'm strategic about the order in which things will exit the bags.
Wow. So $10,000 for winning the championship.
That's not enough. These people deserve more. If you're a champion bagger, you're worth way more than that. But okay.
That's amazing. They have state contests, and then it goes on to the national championships. Let's see. We just missed New York's state championships, which were Saturday, November 5th. Massachusetts were Wednesday, October 26th. Let's see if there's anyone. Virginia was November 5th. Nope. Okay, no more. The 2000. This is all for 2016. Yeah. The national championships are coming up. Oh my God, that was fabulous. They're so serious, right? Salty Ash?
Mm-hmm.
Oh my goodness.
All right. I gotta go.
Yeah,
I do
it's late.
too. And last night was a late night for many of us.
Yes.
So I'm a little tired. I think I need to go to bed. Maybe have another glass of whiskey. We'll see.
Yeah. Okay. But, I need a quick moment with y'all and off-air after show just to discuss a little.
Are we in trouble?
No.
Okay.
What are you talking about?
What? All right, everybody. We will see you next week. Make sure you check out the Science Island hangout tomorrow evening with Ed from Connecticut, scienceisland.org.
And I'm going to try really hard to be there because I think we're going to be ranting.
Rant.
About what?
After the after show.
Did something happen? I was just up watching Netflix last night.
But we will be back again next week, everyone. Thank you so much for spending this evening with us. We so appreciate it, and we hope that you have a fabulous, sciencey week ahead.
Love yous.